So today it is my pleasure to introduce our guest speaker, Dr. Deanna Williams, who comes here all the way from Johns Hopkins University in Baltimore, where she's an assistant professor in the molecular and comparative pathobiology department, so in the medical school over there. And she does some really fantastic work, and we're really lucky to have Deanna over here. She did her undergraduates at Hofstra in New York, then went and got her PhD from Albert Einstein Medical College, also in New York, just a different part. And then she won a bunch of fellowships during her undergraduate and during her postdoc when she moved to Johns Hopkins, where she was awarded a really prestigious K99R00 award from the National Institutes of Health. And she took that award and turned it into this faculty position that she has now in her very awesome lab. She was telling us all about her lab culture earlier this morning, which was really fun to hear about, especially all of the cool activities that they do, and anyone would be lucky to be a mentee of Deanna in her lab. And today she's going to talk to us about her work leveraging the anti-inflammatory capacity of the endocannabinoid system to restore cognitive function in a rhesus macaque model of HIV. But before we jump to that, I want to tell a little interesting backstory. Maybe like a little, not too much of a backstory, but Deanna has over 12 years of training in the martial arts. And when I asked her for a photo for the flyer, she sent me a photo of her flexing, holding a big trophy from the first amateur Muay Thai competition that she won. And I say that because it's really awesome, and also so that you're careful during the question and answer portion of the talk. Okay? All right, wonderful. And then the last thing I want to say is, it's exciting to have outside speakers come in, and present to us, and always talking about some of the cool new stuff, often unpublished work that's going on in their lab. And I think this will be the, hottest off the press data presentation we have, because just 30 minutes ago in my office, Deanna got some new data from one of her students or postdocs and was saying, I'm going to put this in the presentation. I think it's really relevant. So this is super fresh. So with that, I'd like us all to welcome Dr. Deanna Williams, and looking forward to this talk.
Can you all hear me? Is that good? Fantastic. Thank you, Noah, for the invitation and that very kind introduction. I am a lover and a fighter, but mostly a lover, so bring the hard questions on. I won't be upset in any way. These data really are mostly unpublished. The background has some published stuff, but a lot of it's unpublished that literally I got half an hour ago, and I wanted to bring it to get some insight and input from you guys. I'm going to talk about HIV, how it impacts the brain, and kind of my journey to starting this project in my lab. I know the background is mixed, so I'm going to start very, very broad, thinking about how we treat HIV today. So we use these medications called CART, stands for Combination Antiretroviral Therapy. These therapies are designed to inhibit different points of the HIV life cycle. So some therapies, for instance, prevent the virus from binding to your cell. If the virus cannot bind, then it can't infect your cell. I hope you can see that, yeah. Other therapies inhibit really important enzymes, like enzymes that help the virus reverse transcribe its DNA or integrate into the host genome. Again, if you inhibit these key enzymes, the virus can't propagate. And you give these therapies in combination, typically 2 or 3 different drugs at a time. And when you do this, outstanding things happen. You can suppress HIV in your blood to completely undetectable levels. You may have seen commercials on TV talking about this. And so when you measure HIV in the blood, you find when you're diagnosed, you have a lot of virus. When you begin these CART therapies, the virus really, really drops down. In about 12 weeks, you can get to suppression, and you can stay that way, where we can't find the virus at all. So it's really transformed how we think about HIV, and it's helped people's lives dramatically. But it's still important to remember that it is not a cure. If for any reason the therapies are stopped, the virus will rapidly rebound and come right back. So it just puts a Band-Aid on a virus that is ready to go. And because of this, we still have lots of problems with HIV. it remains a major problem even though we have outstanding therapies. And I'm particularly going to talk about one area that's really interesting to me, and it's about the fact that we have an immune response to HIV. Remember, the therapies suppress the virus, so it's always there. Your body knows it's infected. And so because of this, it's going to be mounting an immune response, right, to try to get rid of this pathogen. The problem with this is our immune system is meant to only happen in a certain way. So when you see a pathogen for the first time, you're going to have this very rapid immune response acutely in the first days, 2 weeks, or hours. This dies down, you get a more prolonged response, and then you have a memory and that protects you. The challenge is with HIV, because you can't ever get rid of the virus, this immune response persists into the months and years, and your body gets tired of keep fighting a virus it can never get rid of. And so you move from a very beneficial acute immune response to a deleterious chronic immune response. This chronic immune response is hallmarked by the production of many different inflammatory markers. The names don't really matter. Some are listed here, and you see others listed down below. But the general sentiment you should take away is the fact that when you have HIV, shown in blue, green, and red, these are the 3 different stages of that early acute infection. You can see you produce inflammatory markers compared to uninfected people. And then even when you move into this chronic phase, the markers are still produced to a comparable or maybe even a higher level. And the challenge with this is, even when you begin your CAR-T therapy, shown in the bottom, being suppressed for about 2 years, you can see that these markers, the lines all pretty much overlap, right? There's no big difference. So the virus is quiescent, it's suppressed, but the immune response persists. And this is a problem. Some of the major cells that mediate this are, depending on who you talk to, they'll pick their favorite cell. One of my favorites are macrophages. They're really, really, really helpful and important cells. I think they're really neat because they bridge these 2 parts of the immune system. And they're also really interesting because they're present everywhere. You have macrophages in every organ of your body. And they have general functions. They help take up pathogens and eat them up and get rid of them. That's their general functions everywhere. But they also have unique functions depending upon the organ in which they live to help give each organ its unique role. More importantly, macrophages get messed up a lot, right? They contribute to pathogenesis in many different contexts. Infectious disease, neurologic disease, metabolic disease. Honestly, pick your favorite disease. There's probably going to be a macrophage component. For HIV specifically, they're really interesting because they're the first cells to be infected in your body, which many people aren't aware of. Many people think about T cells as the major HIV source of infection. But if you don't have macrophages being infected, you'll never have infection of T cells. You can also take people who are suppressed with ART therapies and find residual virus, and if you sequence it, that virus always comes from macrophages. And then they can track that virus throughout the body. So macrophages go into different organs and bring that virus with them. And so because of this, HIV can affect, infect, and damage virtually every organ system, and macrophages play a huge role in this because they're in every organ system. I focus on the brain specifically, but you can focus on liver, lungs, kidney, heart. It's gonna be not normal when you have HIV. So let's learn more about the brain and HIV and what happens there. So the brain is really unique. Most people don't know that the brain is highly infected by HIV. And in fact, it's infected within the first 2 weeks of being exposed to the virus. In some cases, you can find it as early as 4 days after estimated transmission. That's really fast. Most people don't even know they have HIV within 4 days of being exposed to the virus, so incredibly quickly. And the virus there is really interesting because it's driven by macrophages and specialized macrophages in the brain called microglia, as well as astrocytes. So these cells have very distinct biology than T cells. They live predominantly and only in the brain. And so the pathogenesis there is a little bit different. The virus in the brain is also compartmentalized, which means it can be genetically unique and specially adapted to survive in the brain. And the brain's a viral reservoir, which means the virus can persist there even when the rest of the body is suppressed. And so if we don't find a way to get rid of virus in the brain, we're never going to cure HIV. HIV causes all kinds of neurologic problems. The ones that I spend a lot of time thinking about are cognitive issues. So the term to kind of summarize this is called HAND. It stands for HIV-associated neurocognitive disorders, and it's a spectrum of cognitive abnormalities. Some of the symptoms are displayed there. Overall, they really impact your quality of life and just your day-to-day activity. I think many of us have walked into the kitchen to get something, gotten distracted and forgot what you went there for, and you're like mildly irritated, right? But this could happen on a chronic level. Or think if maybe you're taking these life-saving ART medications and you keep forgetting to take them. That probably is going to be bad for you, right? So HAND is actually associated with higher mortality than people that don't have HAND. I'm listing here some vignettes of people who have been diagnosed with HAND, some of the things that impact them in their day-to-day. Again, some of these may not seem like major things, but when they keep happening, it can really impact your quality of life. Of particular interest are these here. There's decreased concentration and ability to multitask. Many people have difficulties maintaining employment, especially if their job was something that required a lot of writing and listening and thinking and taking notes, right? Like that's a lot to do. And your brain can't always handle that. And you may think of this comparably to the brain fog you hear happening with people that have COVID. It's very comparable. And so how is this diagnosed? It's diagnosed by neuropsychologic testing. We also do mental health testing to kind of rule out other factors that may be contributing to cognitive abnormalities. These are some of the common cognitive domains evaluated. Different aspects of learning and memory, your ability to process information or process language, and the names of the tests are listed on the right-hand side. So I'll go over a few of these just so you get a flavor of how they're studied. So for verbal learning and memory, you're going to be told these random words that we have some familiarity with, right? And you'll be told, remember these words, they'll come back at some point. And then at random periods of time, they'll say, do you remember those words? And you have to say them. And maybe if you're not from North America, you don't know what a turkey is, right? Like, so that one you might not remember, right? But maybe everyone knows a pencil maybe or a chicken. So it's trying to see how well you can recall these seemingly random words. But I hope you can also appreciate how different people without cognitive impairment may not do well on this test if they're not familiar with these items. So that's a big problem in cognitive testing. Other things we have to do is fluency. You'll be given a letter and have to name as many words you can that begin with that letter. Motor skills, you have to put these little metal pins in a little pegboard with both your dominant and non-dominant hand. Digit symbols, I did this game a lot as a kid. I didn't know it was anything important. But like you have a key and you have to match the key with the letter or the number. Trail making test, you have to go in numerical and alphabetical order. So in A, it goes from 1 to 2 to 3 to 4. You have to connect the dots. Or in B, it's 1 to A, to 2 to B, to 3 to C, and things like that. And then after a while, we compare your results to norms, people that are not believed to have cognitive impairment. We see how much worse you do compared to those people. We rule out any other confounding factors, like maybe you've been getting concussions for the last 10 years. Like that's something that's a problem, right? So we rule those things out. And then you give a scale of essentially the least severe is asymptomatic, which means you have impairment, but it's not enough for you to be aware of in your daily living. Mild is kind of in the middle, and then the most severe is dementia, and this can be likened to Alzheimer's, although it's rarer. And so on average, all these different aspects of HAND impact about half people that have HIV. And that's a big number of people, 50%. And what you can see is that people before therapy and after therapy have an equal chance of having HAND, right? So the therapies do nothing to prevent or treat it. So how does this virus that can cause immune suppression do all these things to the brain? And what do macrophages have to do with any of that? Well, it turns out that monocytes, which are kind of myeloid cell, they're precursor to macrophages. Their job is to go into tissues, look for pathogens, and if they see them, take care of them. So they're doing their job. The problem is they can also be infected by HIV. So if they're infected by HIV, they cross the blood-brain barrier to get into the brain, they can bring that virus with them. That virus can then go on to infect macrophages in the brain, as well as astrocytes and microglia. So now you have infection. Now it's important to realize not every cell is going to be infected, of course, right? And in fact, most cells are uninfected, but they're exposed to a pathogen, which activates them and makes them unhappy, right? This very inflammatory profile. They make a number of different things like chemokines that help recruit more cells into the brain, cytokines that are kind of our inflammatory danger signals, different factors that are meant to help kill pathogens, but in the long term are damaging, and HIV proteins like TAT, G120, and F are directly neurotoxic. So the inflammation, the virus, leads to this neurotoxic environment, and you're going to have a loss or damage of neurons. And depending upon the part of the brain where you have this damage, you're going to have a consequent loss of function. And that seems different for different people for reasons that we don't understand. And so I've become really interested in seeing how these macrophages contribute to inflammation during HIV, and what does that have to do with cognition. And so I'm going to start with some human studies that I've done. We get people that have HIV, they come into a clinic, we get blood and process that to isolate out the immune cells. We also can take the plasma and look for different inflammatory soluble markers. We can take the cells and phenotype them to see what kind of markers they express, and we can also add them to our model of the blood-brain barrier to see how they cross the barrier to get into the brain. We look at different kinds of monocytes. Essentially, humans have two different kinds. They can have one marker called CD14. That is the LPS receptor, so that helps us detect bacterial antigens. And we also can have CD16. This is a marker that helps bind antibody to take up a pathogen. And most people have monocytes, when you're healthy, that have mostly CD14. You see very little CD16, right? Only about 5% of the cells. And there is a spectrum of how much 14 and 16 you can have. The interesting thing about these cells, though, that have 14 and 16, is they're believed to be the ones highly infected by HIV. So they're small in number, but they're the only ones that can be infected. And we can find them in the brain of people post-mortem, and they're positive for HIV antigens. So we think they're the ones that bring the virus into the brain, but we don't really have much evidence at this date because everything is post-mortem. And so we set out to see, are these cells expressed to different amounts in the blood when you're uninfected and when you have HIV? And I hope you can appreciate the fact that they look pretty different, right? These are FACS data, CD16 on the Y, CD14 on the X, and you can see very beautifully when you have HIV, a large number of these cells that have CD16. And this is data from just one person. We looked here from people that have HIV in pink from two different cohorts, and those that are uninfected in blue, and you can see across the board an overwhelming increase in the number of these cells that have CD16. But I think you can also appreciate the wide variability and the extent to which that marker is expressed across different people. So we can take these cells and we want to see how they migrate, because again, these ones are infected. So do they get into the brain? And so to test this, we use our in vitro model of the blood-brain barrier. This model is comprised of primary human cells that we isolate from brain tissue. It's a tissue culture insert, and it has a filter that has 3-micron pores. On the upper side of the filter, we add human brain microvascular and endothelial cells. On the underside, we add human cortical astrocytes. Now, because this filter has pores, the astrocyte in feet can penetrate to make physical contact with the endothelial cells, and this helps seal the barrier to be impermeable to albumin and all kinds of things that aren't supposed to get into the brain. They grow to confluence over 3 days, and at that time, we can use it in a number of different experiments. For these, we add immune cells to the top. They migrate into the bottom in response to chemoattractant. We use CCL2, which is really important for monocytes, and it's also associated with cognitive impairment in HIV. And then after they migrate, we collect the cells from the bottom and see what markers they express. And when we do this, We can look at the cells on the left before migration and on the right after migration, and we can see who's there. For these analyses, I broke up the CD16 positive cells into two different populations based upon how much CD14 they have. So the ones in red have low CD14, those in blue have more CD14. And before migration, you have a mixture of cells, but after migration, there's an overwhelming majority of cells that only express high amounts of CD14 and CD16. Again, this is just one person. We've completed this with almost 30 people, and you can see again, very much the blue cells are the ones that migrate to high extents. And the interesting thing is they do this normally to media alone, and this is enhanced in response to chemoattractant. The other cells minimally migrate, and those in blue are the ones that are infected. So we think they're the culprits that are not only highly permissive to the virus, but they like to cross the barrier to a greater extent, and they bring the virus with them. So we then went on to see, do the number of these cells correlate with cognitive function in people? And they do. We looked at people that had HIV that were virally suppressed, and we wanted to see how many of these cells they had and how many of them crossed. And essentially, the more of those monocytes that were in blue, they can also be called intermediate monocytes. The more of those you have, the worsening cognitive function you have, which is really, really interesting. So there is something about these cells crossing that relates to lower cognition. We also went on to see if you can figure out which domains were going to be impacted preferentially. So we went to look at soluble markers of inflammation that come from monocytes and macrophages in people that were uninfected and those that were infected. We looked at these markers by Luminex, multiplex assay, did very fancy math I'm not going to go into, and we wanted to see how those markers map with cognition. And we looked at different cognitive domains. My logo wasn't showing up, but I can put them. Learning, memory, attention working memory, executive function, processing speed, fluency, and motor skills. And you can see that the markers relating to neuroinflammatory signaling, myeloid endothelial communication, and microglia cell recruitment to the brain were associated with the worsening cognition because they're below this beta coefficient axis. Particularly, certain domains were impacted. The interesting thing about these is that all these processes are related to immune cells getting into the brain. So I think this is further evidence of the role of macrophages and monocytes contributing to worsening cognition. So this is really exciting to us. It's data from people that have HIV, but it's not mechanistic in the sense that I can't track, is this one cell getting into this one brain region causing inflammation, leading to cognitive impairment. And I don't have a way to stop this from happening. I'm just reporting that it's happening. And so we set out to see what can we do to maybe stop the inflammation and to more definitively find the role of these cells in causing cognitive impairment. So we're looking at the cannabinoid system. So cannabinoids can be broken down into different categories, just different kinds. We mostly know plant cannabinoids. These are called phytocannabinoids, and they're present in the cannabis plant, AKA marijuana, right? And so the biggest ones you think of are THC and CBD, but there are many other cannabinoid species in the cannabis plant. Our body also has cannabinoid molecules. They're endocannabinoids, and the classic ones are 2-AG, and I'm blanking right now, 2-AG and AEA. And our body makes these to help us maintain homeostasis. So if you think about the classic phenotypes when people take marijuana, like feeling hot or memory loss or eating a lot, right? The endocannabinoids regulate those systems in us. So marijuana exploits that. And then there's also synthetic cannabinoids. These are small molecules that are designed to look like our own or marijuana's cannabinoid components. And how all these cannabinoids work is by binding to different receptors that signal and tell different cells to do different things. And they're implicated in anti-inflammatory processes and neurologic disease, immune response, viral infection, all kinds of things. And so we think this could be really, really interesting. And in fact, cannabinoids have been shown to have a positive effect during HIV. People have given animals or even humans marijuana. They've given them THC. They've used some of those agonists I was mentioning, and they seem to have benefits. They seem to decrease HIV infection, the amount of virus. They may decrease different inflammatory genes. So this is really, really promising. But the problem is the law. Cannabis is not legal everywhere. Also, not everyone wants to feel high, right? Some people want to, some people don't. And the ligands are very hard to target only to the brain, right? So while these are great evidence, they're not practical or translational to humans. So we wanted to take what we learned from this and see if we can get a better model that we can hopefully go into humans that's legal. And so for that, we wanted to look at CBD. Cannabidiol is the second most abundant cannabinoid in marijuana. It is not psychotropic, so you do not feel high from it. And because of that, it is legal, right? And that's really, really big. Another really great thing about CBD is it is available in a wide variety of preparations that I'm sure many of you have at least seen. You may or may not have had it. That's none of my business. But I'm sure you go to Walgreens or CVS and you see these things everywhere, right? And so you can imagine how nice it would be to take your blood pressure medication, your HIV meds, and have a CBD soda. That would be a very attractive way to get people to adhere to a medication. Another great thing about CBD is the fact that we already have an FDA-approved drug. It's called Epidiolex, and it's used to help a rare form of juvenile epilepsy. And it inhibits inflammation, and that makes the seizures stop or at least improve. And so it's great because if this worked, we already have a drug we can use. And so we wanted to look specifically at CBD to see if it's going to help suppress inflammation from macrophages when you have HIV, and see what that would have the effect of that on tissue damage, or specifically brain and cognition. And so to do this, we're going to be using a macaque model. So these are going to be rhesus macaques that are going to be infected with SIV, which is simian immunodeficiency virus. The reason why we wanted to use this is because the human data are fantastic, but people do all kinds of things that I can't account for. They may take marijuana on their own. They may take other drugs on their own, right? And all those things can impact my findings. They may not adhere to their ART medications, so they have unsuppressed virus. And so if I'm trying to test something, it's nice to have a model without those confounders in the beginning, and then you can go back to humans later. So we use the SIV-MAC251 strain. This strain is really, really important because it infects macrophages and T cells. Some SIV models don't infect macrophages, so obviously we need a virus that does. It's very consistent. This allows us to use an animal with an intact immune system, which is really key. Because when you use mouse models of HIV, you have to change their immune system to become infected because mice can't get HIV naturally. And it's the most widely used model in vaccine studies, so it's great because it's widely used by many people. So this virus is fantastic. It mimics human disease, where you infect the animals and you see high amounts of virus in both the plasma and in the CSF, so there is infection in the brain and the rest of the body. Furthermore, you can use the same ART medications we give people to these monkeys, and the virus suppresses exactly as you would see in a human. So it's a great model. This model is also fantastic because it mimics the inflammation that we see in HIV disease. So this is data from plasma looking at two inflammatory markers, CCL2 I mentioned before, that chemoattractant for the monocytes, and soluble CD163. This is an activation marker of macrophages. And what you can see is that in animals that were before they were infected, or animals that were infected but on ART, have very different levels of inflammation. So we know HIV or SIV increases inflammation, and you can see very clearly that taking ART and suppressing the virus does not decrease its inflammation back to its levels before infection. We also can measure the CSF and look at neopterin. That's another macrophage inflammatory marker. And neurofilament light chain, that's a marker indicative of neuronal damage, so brain damage. And what you can see is, again, is that art is not able to suppress this response that happens when you have HIV in the macaque model. We can also get monocytes from these monkeys and differentiate them into macrophages in vitro and look at them specifically to see if the macrophages themselves are responsible for this inflammation, and they are. We can infect them with SIV, shown here in orange, and suppress the virus with ART in green, and you can see very clearly that SIV produces different inflammatory markers like IL-6, TNF, CCL2, and ART doesn't suppress it. We can also look at these cells. So these are brain macrophages. You have microglia on the top and perivascular macrophages on the bottom. And I'm showing you the CD163 activation marker. And what you can see is in uninfected control animals to the left, SIV alone in the middle, and SIV and ART on the right. You can see that SIV infection increases its activation marker on macrophages. You see a lot more red. And with ART, it's equally, right, as expressed. So again, this is a great model because it seems to model what we see in people. We can also not just look at the macrophages, we can get them out of different organs. So we can do enzymatic digestion to get them out of different tissues. So this is a spleen. And I'm showing you FACS data showing that we can get purified cells that have certain markers. This one is MMR. And we can also get macrophages from the brain that include both macrophages and macroglia, shown here in this plot. These others are just controls to make sure we're not getting the wrong cells. And we can profile these cells to see the genes they express. And so we look at animals that were uninfected, infected with SIV, or infected and suppressed with ART. And the names of these genes don't matter, but they're related to inflammatory or antiviral signaling. And you can see that SIV infection increases the level of these genes, and ART doesn't reduce it. And in some cases, like this one, for instance, it makes it worse. So further telling us that the model is great, and we have a really big problem on our hands in macrophages. And so what we wanted to do in this model is to see how CBD impacts this macrophage immune activation, all these different inflammatory markers that I've been talking about. We wanted to see how it impacts the antiviral signaling that promotes inflammation. And then we also want to see if we can find some way to look into the brain in people that are alive, because you can't get brains until people have passed away. And so we want to see if maybe we can get monocytes from blood and find something in them that's comparable to what's happening in the brain and microglia. And if that's the case, we can easily take a blood sample and have a peek into the brain. So we're trying to identify some markers to see if they're comparable between the two cell types, and maybe CBD is going to impact them comparably. And so for the study, we're going to take our macaques, we're going to infect them with SIV for 2 weeks. After that time, they're going to get an ART regimen shown below. This is a first-line regimen given to people that have HIV, and they're going to be suppressed for 6 months to ensure we have really, really great suppression of the virus. Along this time, we're going to take blood and CSF throughout to profile the immune cells, look for inflammatory markers, and then at the terminal time point, we can perfuse and collect organs of interest. We're going to do this for different experimental groups. We're going to have controls, of course, uninfected animals, those are uninfected and getting ART, those that are infected or infected with ART, and then, of course, those that get CBD. And we also have to have a special group, a pharmacokinetic group. There hasn't been published studies looking at CBD in macaques yet, so we didn't know how much to give them. And people ingest all different amounts, so we want to make sure that what we're giving is going to be comparable and a safe amount that could maybe go into humans. When this study is done, what we're going to do is get different organs. We're focusing on the brain, of course, but I think it's really important when you're doing animal research to respect the animal who sacrificed their life for us, right? And get everything. So we're getting every organ because macrophages are everywhere. And so we're going to be looking in adipose tissue, liver, spleen, lung, blood, everything. We're going to do tissue digestion on the solid organs to get the cells into a liquid suspension. On the liquid organs, quote, like blood and lung lavage, we just do a density gradient isolation. Thankfully, macrophages have a common marker, C11b, it's an integrin. Now, not every macrophage expresses this marker, but macrophages, at least some of them do in every organ, and so that we can at least get some of them. But it's important to note we will be missing some macrophages because, again, they're specialized for each organ. And then we'll profile them and look at all different kinds of things, evaluating their inflammatory status, how infected they are, their antiviral genes. But we're looking at some really important macrophage functions like phagocytosis or reactive oxygen to really see what's happening and getting at the crux of this inflammation. And once we have an exact idea of how the inflammation is happening and if CBD is going to stop that, we can tie that back to cognition, because monkeys are really, really smart, right? And so we're going to it's called the CANTAB test, Cambridge Neuropsychological Test Automated Battery. It's essentially an iPad-based neuropsych test for the monkeys that you train them on. And so you can pretty much give them a reward, food, because we all love food, right? Teach them how to do this test, and it's just a point and tap. And it tests key domains that we look at in people. And so it's great because we have a really great correlate of a human cognition. This is what it looks like when it's in the macaque. So you have to have it, the animals are of course in their cage, and then through the cage they can poke their finger through and tap. And so it's a really great mechanistic way. Once we know if CBD is doing something or not, you can do experiments where you, for instance, deplete a certain macrophage population, and once that macrophage is gone, you can have definitive proof, quote, that they're contributing to the cognitive delays that we're seeing. This is ongoing. We just started, so I'm super excited. I will come back with updates and let you guys know. But what we're doing right now is we're working on our PK study, because again, CBD's never been published in the macaques. And so what we're doing is we're doing essentially a 2-week study. We're taking doses that are given in behavioral studies to rodents, doing math to make them adjusted for macaques based on their weight, and we're doing escalating doses followed by washouts. And so they're going to get a low dose for a few days, stop, let them recover, a higher dose, stop, so on and so forth. And then we're going to be collecting urine and blood for many, many, many intervals to see how much is there. At the end, we're going to be getting organs and practicing getting on macrophages. But this is also great because we can get a really good glimpse into how acute short interval CBD, what it may be doing to the macrophages. We also think it's important, I didn't mention earlier specifically, these studies are going to be done in both males and females, which is really, really important because women are half the population, of course, but also female macaques are missing from a lot of studies because they're used mostly for breeding, which is really important. But because of that, we're missing out on key information about females and how that impacts the immune response. And so this matters because we found that in humans, there are differences in CBD metabolism. So this is data where you give people oral CBD, you measure how much is in their urine for a number of days. And this is a CBD hydroxy metabolite. And what you can see is that, really interestingly, females have a very early peak CBD concentration, right? But males have a higher absolute concentration. It just takes longer to happen. And so we're trying to make sure our macaque PK resembles this as best we can, and we'll pick a dose based on that. Further in the meantime, we're going to be looking at some of these endocannabinoid receptors. So remember I mentioned cannabinoids work through receptors to cause these inflammatory benefits. You have different ligands. You have enzymes that make them up and break them down, and they work together to have the inflammatory effects or anti-inflammatory effects. And CBD takes advantage of this. We want to look at these receptors because CBD has a really complicated pharmacology. It can interact with a lot of receptors, a lot. And so because of that, if we see any effect, we won't necessarily know who's involved. And so we're trying to get a glimpse of this because since it can't interact with so many receptors, maybe we can make one particular ligand that activates just one instead of all 10 receptors, for instance. It can activate some, it can inhibit some, it does a lot of stuff. And so we've been looking at these receptors in the brain, and we've been looking at them across different brain regions, which we think is really important because the cognitive abnormalities are restricted to certain brain regions. And we found really cool things. So the brain regions are at the bottom. These are 3 different cannabinoid genes, CV1, PPAR alpha, TRPV2. The latter 2 have additional other functions beyond cannabinoids, but they also combine cannabinoids. And what you can see quite broadly is that there's differences, right? CV1 is the most highly expressed, but it's not expressed equally in the brain. And I think that's really interesting and something we need to think about. Others are present, but they are also heterogeneously expressed. And this X indicates that I didn't have sample for that one, so that one wasn't run yet. We've also been looking at this in different species, which we think is really important. We've been looking at this in mouse as well. Not everyone can do macaque studies, and most cannabinoid studies are done in rodents. And so we want to see how the macaque applies to the rodents, because that's what most people work on. And it's interesting, they are different. So here's the macaque I showed you earlier, and here's C57 Black 6 mouse. And it's interesting. So CV1 still has heterogeneity, but it is a little different. And the other 2 are pretty much equally expressed, except for this one in the hippocampus, which is for whatever reason low, right? So for these genes, the mouse doesn't have the complexity that the macaque does. And I don't know what it means, but it certainly could give some reasons why we see differences when we go from preclinical models up to making therapies in humans. We've been looking at this outside of the brain too, in the periphery. You can see different peripheral organs there. Now we also have rats. And so you can see essentially differences. Again, the X means I just didn't have that sample at that time to run it. And so you can see that some things are different. In macaques, CV2 is highest in spleen, but it's there, right? But it's not as highly expressed. And then the rats, it's kind of equally expressed everywhere with nothing standing out. So there are differences even between rats and mice. And again, I think these are noteworthy, even though I don't fully understand the implications just yet. And here's another receptor just to kind of drive it home again. This is Fa, an enzyme that helps metabolize cannabinoids. And again, you can see kidney has the most in macaque, but liver is the highest in mice, and colon's the highest in rat. So if you're making a therapy or trying to make a therapy, you need to think very carefully about the animal model system that you use, because not everything is expressed equally across different species. And so with that, I just want to summarize. At the very least, I hope you know macrophages are everywhere, and they're important, and they contribute to inflammation during HIV. And while we have therapies to suppress the virus, they don't do much for its inflammation. And it's a big problem because it makes the brain vulnerable to cognitive and other abnormalities. And we're hoping that perhaps CBD could be a very great adjunctive agent to help suppress inflammation that is legal, widely available, and you can just take a gummy bear and suppress your inflammation. And so with that, I have to thank my lab who has done all this work, including Hannah, who just joined, who doesn't have a body yet. I can't Photoshop, but she's real, I promise you. And Twitter and email and acknowledgments and money, of course. So I'll take questions you may have at this time, and thank you for your attention. Yeah, please.
So I have a basic question about the persistent inflammation. So do you think that is induced by undetectable levels of viral replication, or is it that these cells just become permanently inflammatory irrespective of
Could you repeat the questions?
Yes. So the question was wondering about where this inflammation is coming from. Is that these cells are responding to low-level virus that's still there, or is it because they become essentially hyperactive consistently, right? I think it's both. So we know HIV integrates into your DNA. It always is there. always, even when it's not making virions. And so your cells have intracellular means to detect foreign DNA. And so they know they're infected. And so likely that's being a trigger for an immune response. Also, cells, when they become activated for so long, they become dysfunctional, broadly speaking. And that dysfunction can persist and sometimes can't be repaired. And the thing about macrophages is they're very long-lived. So they're going, especially in the brain, they're there for decades. And so once they get turned on, they could potentially stay on forever. Another really important thing I didn't talk about, but the other half of my lab's focus is on these CAR-T therapies. Because we have a blood-brain barrier, which immune cells can cross beautifully if they choose to, but therapeutics cannot cross, including ART therapies. And so the other half of my lab looks at these therapies, how to get them across the barrier, what transporters they use, are they neurotoxic? And so I think another big source of inflammation is the fact that these ART therapies just can't get there, right? So if the virus is in the brain, but the therapies can't get in to suppress it, it's also just hanging out, kind of wreaking havoc. So it's a lot of features, but great question. Yes, please.
You talk a lot about the variability of cannabinoid receptors between organisms. Have you looked at the variability within one organism? Because I know at least in humans, like getting high feels different for everyone.
Yes.
So I assume that the cannabinoid receptors would be expressed differently in the brain and the cells and the organs and stuff between people.
Great question. The question was thinking about interdonor variability within, across one species, right? Because the different species look different. And it's true, different macaques do look different. Different mice do look different. And I didn't show it because the data were kind of averaged out, but mice were less variable. Macaques had more variability, which is really interesting, right? If I could find a way to put error bars in a heat map, I would, but the mice were really, really similar. The macaques had more variability, not as much as humans, right? Because the human spread is like this. But macaques do have variability, and I think that's another reason why things kind of fail as you move up the clinical model systems. Yeah. There's a question over here, I thought. Yes, you had your hand up.
I'm not sure about the legality of like terpenes, but have you looked into anti-inflammatory terpenes and maybe possibly a synergistic effect?
Yeah, the question is about terpenes, which is another really important cannabis component. I have not, but I'm interested. I mean, CBN, CBG, I'm interested. And once we figure this out, because the thing is, if cannabis has like 100 different components, it may not just be one individually. Likely, it's some of them working together. And I imagine because they all interact with different receptors, the signaling that happens with one alone is different when you have multiple things together. And so you could imagine a place where you can kind of, not maybe for all 100, that's too much, but you could titrate in different amounts and maybe hold on the pharmacology. So yeah, I want to move more broadly, and that'd be really cool to see. Yeah, thanks. There's a question over here? Yes.
As you show, there's a bunch of different ways you can take CBD. I was wondering how you were administering to the macaques and how that might affect its effect or its effect on.
That's a great question. The question is about how we're dosing the CBD. So we're doing something very novel, but I think, of course, I think it's great, right? But I think it really is great. So most CBD studies in rodents do IV, so they intravenous injection, which is fine. It's super consistent, right? You know they're getting it. Some people do vape, which I think is very applicable for a lot of people. But for a therapeutic, I don't know if everyone's gonna want to vape. And I don't think people are gonna be taking IV therapies at home, you know? So we're doing oral administration because I like to eat and I think most people like to eat. And so CBD is very, very hydrophobic and lipophilic. So we're taking CBD, dissolving it in peanut butter oil, putting that in peanut butter, and giving our macaques peanut butter graham cracker sandwiches, which I think they'll be happy to get their CBD every day. So yeah, good question. I mean, if you can get them to stay still. People that do rodents do vape, but so let's be clear, they don't have like a, you know, only because they don't have thumbs, right? But you can't put them in a little box, and you just kind of like gas them, and they do kind of like go to the gas. like it's really interesting, like the behavior aspect. I'm not a behavioralist, but like we have vets that do it, and we're just gonna like videotape them because I really want to understand, like, do they really like this, you know? But so the macaques, maybe they could vape, but we'll just get them to have a snack. They do have thumbs. Macaques have thumbs. Yes. Oh yeah, follow up. I'm sorry.
The second part is like, I think the mode of administration.
Oh, yeah, you're right. Yes. So, route of administration, yes, it definitely impacts things. So, the pharmacokinetic profile changes based upon how you give a drug. And that's any drug. And that's why we want to do oral, because we think that's how it would go in people. The FDA approved drug is an oil that people just like either put in stuff or they just kind of swallow it. So, we want to go oral because I think that's how people take it. I'm not going to say that one is better or worse, but the metabolism is different. When you take something, it goes right through first pass metabolism in your liver. If you do IV, it goes other places, and then that happens. And so the amount of CBD you have or the amount of metabolism that occurs that's going to break that down does depend upon how you give the drug, and that's something that really is important and needs to be considered. Yeah, there's a question in the back. Yes?
This might be silly, but you hear a lot with THC, like building a tolerance
Mhm.
or something. Is that just associated with the head high? Do you see similar things with CBD? And does it still, no
Yeah.
matter the dose, affect inflammation the same way?
Great question, and I don't know. The question is about tolerance, wherein for THC or maybe many illicit substances, right? You hear people starting off at some amount, and then maybe they're going to take more, and then they're going to take more. I'm not familiar with the literature to know if that exists for CBD. We're trying to look at that to some extent in our PK by dosing up, right? And we're going to be taking blood along the way. So we can take half for plasma to measure CBD, but we can also look at the immune cells and begin to see if any of their other markers change throughout this dosing. We can't get organs until the end, unfortunately, so I can't assess that. But it's something to consider for sure. Yes, please.
You mentioned a couple of complications at hand are inability to think or inability to retain employment, but also that in this sort of research, the biggest problem is the law. So what would you say to someone who is experiencing these complications and trying to manage their condition now? Is there anything they can do?
Yeah.
Substances are more widely accepted.
That's a great question. The question is about the law, and you're trying to get me in trouble. Marijuana is federally not approved for recreational use, and so that is my answer. But CBD is legal, right? So if that's the case, I would say think about how you're going to get your CBD. One of the biggest problems with CBD and all these other kinds of things is that they're not regulated by the FDA. They're legal, but they're not gone through a similar process. And so you can kind of like call it whatever concentration you want, and no one is holding you accountable for that concentration. You don't know exactly how pure it is. So I think the problem with recreational use of anything, honestly, is that there's not systematically done like a prescription is that you get from your pharmacist, right? And so I think if there was a way to make sure it was a standard dose and a standard route, it might be helpful. But I wouldn't advise just taking one you can get from anywhere because it's not going to be monitored, it's not going to be consistent, which means if there are any effects, it's not going to be comparable across people or even you, because different batches are going to have different potencies. And people are even looking to see how much CBD is in these things. And let's just say there's some false advertising. Maybe it's there, but it's not to the extent that the gummy bear, let's say, is saying is there. People are recommending for all kinds of neurodegenerative and cognitive stuff, different things. Diet is one, that's super common. Going to a more plant-based or ketogenic diet, exercise, music therapy, art therapy, all these things are prescribed in different populations. And I think the problem with HIV is there's not as much awareness and importance to like infectious disease doctors thinking about this, so people don't tend to get anything that might help them preserve cognitive function. Yes?
You may have answered this, but does the amount amount of time a person infected with HIV have an effect on immune responses? Like, is there more inflammation?
Great
with
question.
More increased time, or is it just
Yeah.
until it fails, or what?
The question is about how long you're infected with HIV and then what that has to do with the immune response. And it's not linear. It's all dependent upon the person. So what I'll say is, just like anything, think about COVID or a cold. Some people get a virus, right, and they're like breezing through life. They get sick for a day and they're fine. Other people get the same bug and they're like at home for a week. It's just different people are different. It's more about the person. And everyone has a normally natural different amount of baseline inflammation, right? Some people naturally have lower, some people naturally have higher. And so when you bring a virus into it, that's just kind of exacerbated. So it doesn't have to necessarily do with how long you've been infected. But it is known that if you became very immune compromised before you got art therapy, you tend to do worse because your immune system was really, really kind of ravaged, right? And so it's hard to recover from that level of depletion. Even when the cells come back, that memory kind of persists. Yeah, there's a question in the back, I thought. Yes?
This is very interesting research. Would you say it's possible to use this same approach, the same model to approach different viral infections that also affect the neurocognitive system?
Yeah. The question is about, could we use this model to look at other viruses that impact the brain or cognitive systems? And totally, right? I don't know if macaques can get every single virus that we do, but certainly you could. They were really, really integral in helping make COVID vaccines, so they can definitely get COVID. And so, yes, I think it's really, really nice to have animal models to tease these things apart. I think it's important to go back to humans too, because that variability is so key, and I think kind of integral to why things fail sometimes, but they're useful for helping us do things like take longitudinal sampling, take out every organ, those kinds of things that we need that foundational information. And it's not just viruses, other bacteria and fungi and all kinds of parasites, pathogens cause CNS complications. And so I think broadly it's really, really interesting. Yeah. Yes.
I'm sorry if I missed this. Did you have or show any in vitro data showing the effects of CBD on macrophages?
Yeah.
and immune cells?
Great question. The question is about in vitro data, looking at how CBD affects macrophages. I did not show those data. We're working on it. And we're using human cells for this. And it's interesting because it seems to vary upon the person. We're having technical problems because we're using a standard dose of CBD. And some people's cells respond well, meaning we see decrease in inflammatory markers. Other people's cells die, which is bad, right? And it's the same aliquot. So we do experiments with 4 donors at a time to make sure that we're kind of biasing ourselves to get the best data we can. So the same aliquot of CBD, which means that probably some people's cells are particularly vulnerable or sensitized, and they probably need a lower dose, right? But because we haven't worked out what that is for every person yet, or why that happens, it's kind of hit or miss. So it's like, oh, they alive? Oh, great, you got, okay. Oh, they all died, okay. So we're trying to figure out what that is, but I think that's important. And whenever the time comes that we standardize this and get this working, I think it's important to publish those kinds of data, the quote negative data, because at the very least, other people aren't gonna reproduce it and kill other cells. But I think even more importantly, it tells us something about people, right? That this, you may need to think about dosing, because one dose might not work for every person, could even be damaging for some people. So we're a work in progress, but hopefully soon. Oh, yes.
Question, and I may have missed this, but you were showing it in terms of the population of people that develop hands, it's quite a bit, almost 50%. Are there risk factors associated with, there's a certain group that's more likely to get it, or is it just dependent on how many intermediate macrophages they
Yeah.
have, and is that?
Great question. The question is about hands, and vulnerability to getting hand and if there are any risk factors associated. Sadly, at this point, we don't have much. We have no idea why some people get hand, why they don't, and what makes it go from asymptomatic to being like dementia. We don't know any of those things. We speculate. And some of those are, an interesting one is thinking about cognitive reserve. And this is kind of like people's inherent ability to tolerate stuff, right? To tolerate bad things happening. And perhaps some people that have lower cognitive reserve are more vulnerable. There are things that seem to be protective, like higher education. That seems to be associated with less chance of having hand. And I think that kind of makes sense. You're working your brain more and things like that. But I think it's also challenging because lots of social factors are known to impact cognition normally anyway, like diet, or if you had a traumatic upbringing, right? Or like those kinds of things impact your brain and certainly impact the development of your brain. And I think part of the reason why we can't figure this out is people have all different kinds of childhoods, for instance, right? And we all have different amounts of emotional baggage and, you know, all those things. We grew up in different environments. We have different stressors. Our parents, our moms may have different infections when they were pregnant. All these things impact your brain. And of course, we don't have a record of those things. So then you kind of carry those on and grow up and get HIV, let's say, and you carry that memory with you. And so our brains are all vulnerable in different ways that we haven't fully figured out. But I would love to be able to contribute in some way to that.
Is there any difference globally in terms of this type of information available in other countries, or have you seen a difference?
Great question. The question is about global incidence. It's really hard. And a lot of it's not biologic. If you remember when I was mentioning cognition, like the things on that list, those are not globally relevant, right? And so everyone has clocks, or most people have clocks, right? But like not everyone has certain things on that list. And so when people were doing these tests in other parts of the world, they're finding like, whoa, everyone has dementia. But then they're testing quote unquote healthy people, and they also all had dementia. And they realized they had to make culturally sensitive tests. And those tests require you to know the culture, know the language, you know, and that's hard. But now that you're getting a little more awareness is there, so it's getting better. And once we have better tests, and also we're normalizing to people from that same country, right? We're not normalizing to someone here in California, we're normalizing to someone from that same area. And I think then we can get better incidence. But it's prevalent worldwide now that we have better tests. But awareness is lacking, even more so than here. And it's important. And I think there's also different stigmas in different countries with anything related to mental health and kind of cognition and perceived craziness, right? Like, and so that can be a whole big thing. But people are aware, at least clinicians and researchers certainly are. So there are studies, big ones are in Thailand, Uganda, South Africa, Germany, Italy, all over, but there are a lot of them. And so people, Brazil, Puerto Rico, so they're trying to kind of get a sense for this to see. But yeah, you need a good test in that language, and it needs to make sense to them. Yes?
One more, sorry. So currently you're getting the brain samples from deceased donors.
Mm-hmm.
Do you think in the future you'll be able to test macrophages and see this in living donors? Or is that's
Ooh,
far?
great question. The question is about, we get brain from deceased people, of course. We can't take people's brains. Could we get macrophages in people that are healthy? We can. Blood we can always get. That's really easy. They're starting to now do transplants, organ transplants, from people that are HIV positive to HIV positive, which is awesome for us. This is really, really, really new. Hopkins did the first one ever in the world like 2-3 years ago. And so sometimes if you're link up with the clinician, you can get a piece of that organ, the person consented, of course, right? And you can get macrophages from there. But you have to like know the person that's doing the surgery and know the surgery schedule. It's complicated. So you can. Because, I mean, and even that has complications because it's not healthy. Yeah. There are also times when people get I forget. You take out some organ. I don't know which one. You can survive without it. Maybe gallbladder. I don't know. And when they do that, they do a partial splenectomy sometimes, and that spleen is healthy. They just do it for reasons that I don't know. And sometimes we can get those spleens. So that's really cool because it's a healthy spleen, right? But in general, macrophages are hard because they're in organs and we can't just, I mean, that's like a sci-fi movie, right? Like we can't get people's organs, and that's why we need animal models to kind of study this.
Thank you.
All right.
So let's all thank Dr. Williams.